The last thing that we talked about was what happens when glycolysis is over. And so I just want to recap aerobic versus anaerobic respiration and perhaps add a couple of details to it. So you already wrote down a lot of this information yesterday, if you were writing things down yesterday. So before you write it all down again, maybe take a look at what's there. I just want to remind you that anaerobic respiration is when glucose is turned into lactic acid or alcohol. And we can sometimes call this fermentation depending on which type of cell it's happening in. Now, anaerobic respiration for us happens in muscle cells. We saw that yesterday, when strenuous exercise is happening. And the thing that I maybe want to add on here is this point here. What's really happening is not that you have no oxygen available at all. It's actually that your cell is using oxygen faster than your circulatory system, so then your blood can provide it to you. And not all cells can do this. So I wanted to mention quite specifically that a cell in your brain doesn't do this process. It is unable to perform anaerobic respiration. And so some cells in your body don't just hurt without oxygen, they die without it. So it would be realistic for me to ask you a question where I say, I have a brain cell and a muscle cell, where could I have anaerobic respiration? So knowing that difference would be important. Side note, does anybody know what it's called when your brain doesn't have oxygen? There's like a real word, like a term for it. It's fancy and sciencey. Asphyxiation is close. That's when you can't breathe and thus you have no oxygen. But you're on the right track. There's a word that means that you don't have enough oxygen in your brain. So I'm just going to tell you, a stroke can cause this. The word is ischemia, cerebral ischemia. I'm not even joking. Every single person who has ever died has died because of this. Ever. I'll tell you how. Let's say that you die because you have a heart attack. What actually happened? Your heart stopped pumping blood, your brain doesn't have oxygen, you die. Let's say that you get your head chopped off. What killed you? Your brain's not getting oxygen, so you die. If there was a way to continue feeding your brain with your circulatory system's products, the oxygen that's in your blood, your brain could stay alive. The rest of your body, of course, is detached. That would be a problem, but your brain could keep living. If you drown, what happened to you? You couldn't breathe, so your brain had no oxygen, so you die. If you got shot by a gun, what happened? You probably bled out, so your circulatory system couldn't bring oxygen to your brain, so you died. This is what killed everybody, ever. I'm telling you. Yeah. If we're talking about human people, for you to have been alive, you had to have been acquiring oxygen somehow. It doesn't mean that you breathe through your mouth. It could be that you were attached to a machine that was breathing for you. But if your brain cannot get oxygen, you die. Simple as that. If you die from an infection or a disease, well, let's think about what the cause of this is. If you have an infection, what is the issue? Probably your blood's all mucked up. So usually if you have an infection, you have a bunch of stuff in your blood, preventing your blood from carrying oxygen to your brain, so you die. It could be that the infection causes one of your organs to fail, and that prevents your blood from carrying oxygen to your brain, so you die. That could be the reason. If you die from a disease, so let's say that you are dying from, I don't know, pick a disease. What's that? So you're dying from AIDS. Now, what actually happens with most people who die from AIDS isn't that AIDS is killing them. It's usually that their immune system cannot respond to some other thing, like, let's say, pneumonia. So you have pneumonia, and your immune system is unable to attack it, so your body is unable to clear all of the mucus that's in your lungs, so you can't breathe, so you die. I'm telling you, everybody who ever died, this is what killed them. Side note, I have a degree in neuroscience, which means I spent 4 years doing nothing but studying about brain stuff. So if you want to ask me about brain things, I actually know a lot. The thing that I told you yesterday was this. Lactic acid decreases pH, so muscles cannot contract. Now, how would you overcome this? And we're going to talk about your muscles in the next section. But just as a preview, the way that you overcome this is to take your oxygen debt, which means your lack of oxygen, and meet it by increased breathing and heart rate. So why do you breathe hard when you exercise? Because your body is demanding oxygen, and you are responding without even knowing to provide it quicker. Why does your heart beat faster? Because your body is demanding oxygen, and your blood needs to flow quicker to provide your cells with oxygen. As this happens, lactic acid is removed. And one of the things we're going to do in this section about muscles is read a couple of articles to maybe debunk a myth that a lot of people have. I'll tell you, for example, my legs still hurt from this ridiculous workout I did at the gym on Sunday. They still hurt today. I don't even want to walk up and down stairs. And the common misconception is that I still have lactic acid in my legs. Incorrect. Lactic acid gets washed out as soon as you start breathing fast enough to replace the oxygen that was missed. And so one of the things we're going to talk about is, why do your muscles actually still hurt 2 or 3 days after you've exercised? Because the answer is not lactic acid. The thing that I'd like to point out here, because we're going to talk about ATP today, is that the net ATP that you get from anaerobic respiration is 2. 2. Not 36, like I said, you can get in optimum conditions in an animal cell, just 2. Is there anything that anybody would like to ask about anaerobic respiration here? Oh yes, and we're going to talk about this, but I've heard lots of people, not just your age, but like grown adult people. I've even heard people who claim to be professionals in the industry claim to me that if I get a massage, what I'm doing is getting rid of the lactic acid in my muscles, which is completely not what's happening at all. And so you're right, and we're going to just talk about that a little bit further when we do the muscle section. This is just a preview to it. So here's what we need to talk about then. We need to talk about what happens when I do have oxygen. So if I do have oxygen, here's the next step. Oh, let's make this the right size. If I do have oxygen, what happens next is what we call the link. Something has to happen between glycolysis and the Krebs cycle before I can continue on. I alluded to it before, but here's what's happening. Glycolysis is the first step, happened in the cytoplasm. Before I can move on and go to the next step, the Krebs cycle in the mitochondria, I need to take pyruvate, and that little 3 refers to the fact that there are 3 carbons. And pyruvate undergoes a reaction with something called coenzyme A. And the result of this is actually a couple of things. The result of this is that I get acetyl coenzyme A, which is sometimes just referred to as acetyl-CoA to make it a little shorter. This 2 is referring to the fact that there are now only 2 carbons. So I have essentially taken off a carbon from my molecule of pyruvate. Now if we take a look over here, here's my other products. I get a molecule of CO2. The process that caused this is called decarboxylation. D means I undid something. Carbox talks about carbon and oxygen. And ylation means I took it off of my molecule. So decarboxylation is saying I took carbon and oxygen off. The other process that happened was oxidation. I oxidized pyruvate so that I could reduce a molecule and get NADH. So just so we're clear, pyruvate is what got oxidized. NADH is the reduced thing that I made because of that. So pyruvate gets oxidized, pyruvate gets decarboxylated, and the result is that I have acetyl coenzyme A. It has 2 carbons instead of 3. And so down here I have the text that basically says what this reaction is showing. Once pyruvic acid or pyruvate is formed, it enters the mitochondria. When inside the mitochondria, I take my pyruvic acid or my pyruvate, which has 3 carbons, and I convert it into a new 2-carbon compound called acetyl coenzyme A. For that to happen, I remove carbon dioxide, which is decarboxylation, and I also take hydrogen. This we call the Link reaction. It's the thing that happens in between glycolysis and the Krebs cycle. So I'll just wait here and give everyone a chance to write stuff down. Maybe I'll just pause the old video. So step number 2 is the Krebs cycle. Another name for the Krebs cycle is the citric acid cycle. I have the tendency to call it the Krebs cycle, but it can be referred to by either name. We'll talk about the text that's on the right first. You'll notice that I've got a pretty detailed picture here, but it's because I'm trying to highlight just a few parts of it. So before you try drawing the whole entire thing, let me show you the overview, and then you can see what it is that you actually need to write down. The Krebs cycle is cyclical, so it's just like the Calvin-Benson cycle where there's technically no beginning and no end. The idea is that acetyl coenzyme A enters the cycle. In photosynthesis, CO2 entered the cycle somewhere. Here, acetyl coenzyme A is going to enter the cycle. It will be added to something that has 4 carbons to make something that has 6 carbons. So notice, I'm not talking about the names of all of the compounds. I'm talking about how big or how small are they. If you're interested, I could tell you the names of all of the compounds, but it's not the main focal point of our discussion. So I take acetyl coenzyme A, which we saw had 2 carbons, and I add it to something that has 4 carbons so that I can make a 6-carbon compound. Then, this 6-carbon compound is broken down so I can get the original 4-carbon compound back. So if I compare this to the Calvin-Benson cycle, in the Calvin-Benson cycle, CO2 enters and joins that molecule, RuBP, and then eventually I get RuBP back. Same thing is happening here. Acetyl coenzyme A joins the cycle. It's added to this compound that has 4 carbons, makes something that has 6 carbons, and then eventually I get the original 4-carbon compound back again. In the process, I get some carbon dioxide molecules, I get some NADH, and I get some FADH. Now technically FADH is FADH2. There's two hydrogens. And I don't like to focus on that because it makes people think that something has to happen twice. FADH and NADH are just reduced molecules. They didn't have hydrogen before, and now they do. Just like the first step we talked about today, the link, for every one glucose molecule, this cycle occurs twice, and it occurs in the matrix of the mitochondria. So I'd like to highlight some points of this cycle, and these are the points that I would want for you to know as well. So you'll notice I'm not going to ask you to know the names of the compounds, but simply what's happening to them. I start up here with acetyl-CoA. That's probably the only name you really need to know because it came from the step before. It has 2 carbons. This molecule over here, which is called oxaloacetate if you're interested, has 4 carbons. So you can see in the picture there are 1, 2, 3, 4 little boxes. The idea is that those 2 carbons and those 4 carbons make something that has 6 carbons. And then, over a number of steps, that 6-carbon compound is going to get oxidized. Now, do I need you to know each step and which order they all happen in? Absolutely not. I need you to know that after I get this 6-carbon compound, I start oxidizing it. I get, for example, a molecule of CO2, and then another molecule of CO2. I get NADH being produced. I get another NADH being produced. I get a molecule of ATP. I get FADH2. And I get another NADH. So when we think about this cycle, I don't want you to think about, okay, first I get a CO2, then I get an NADH, then I get another CO2, then another NADH. The order is not what's crucial here. What's crucial is that you recognize that I took this 6-carbon compound, I oxidized it, and in doing so, I came all the way back around and had my 4-carbon compound back. While I oxidize it, I get a whole bunch of substances being produced. If you take a look, you'll see that there are 2 CO2s. You'll see that there is 1 ATP, that there are 3 NADHs, and 1 FADH2. And it's that total number that interests me the most. Do you know how many of everything I'm producing? Not in which order am I producing them. Would anybody like to ask any questions about that idea? Yes. So what I'm saying is you don't need all of these steps of the diagram. You need the general overview. So I'm going to sketch it out again without all of the details so that we can see just the main idea. The main idea is that I have acetyl-CoA. It joins something that has 4 carbons, and they make something that has 6 carbons. The names of those other compounds are not things that you're going to have to know. We're just talking about how many carbons. Then this 6-carbon compound is going to get oxidized and eventually turn back into the same original 4-carbon compound. And while it does that, I'm going to get some things being produced. In no particular order, what am I producing? 2 copies of CO2, 1 copy of ATP, 3 copies of NADH, and 1 copy of FADH2. This is what you need to know about the Krebs cycle. You don't need to know the names of the intermediate compounds or the exact order that all of those things are produced in. You need to know the process. I take this, I oxidize it, and in oxidizing it, I make a bunch of other reduced molecules, and those are the products of the Krebs cycle. Now, I would like to mention that this happens twice. It happens twice because I start with acetyl-CoA, and I know that that happened twice because after glycolysis, I had 2 copies of pyruvate. So the idea is that every time cellular respiration happens, this cycle happens twice. Would anybody like to ask any questions here? So I think what I'd like to do, we just have one more real slide of notes left to talk about, and I'll save that for another class because I'd like to talk about the lab that we're going to do tomorrow.